So the first question we asked was to determine whether or not pentamidine did in fact synergize with gram-positive active antibiotics. To do this, we conducted a checkerboard assay using pentamidine in combination with rifampicin. So in these assays, we're looking at a matrix of different drug concentrations, looking for interactions between the two molecules. Dark blue represents 100% bacterial growth, and white represents bacterial cell death. So you can see that in this assay, as we increase our concentrations of pentamidine on the x-axis, the concentration of rifampicin required to inhibit cell growth decreases with increasing pentamidine concentration. This leaves us with a staircase. This leaves us with what we refer to as a staircase in our checkerboard assay, which typically means that these two compounds are acting synergistically. We tested some additional gram-positive active antibiotics such as novobiocin and erythromycin. And in E. coli, we once again observed this staircase phenotype in our checkerboard assays, telling us that these compounds are acting synergistically. So we hypothesized that pentamidine was somehow disrupting the outer membrane. One of the first assays we conducted after observing our potentiation was atomic force microscopy on E. coli treated with and without pentamidine. Here we're looking at the surface topology of the cells. And in particular, what I want to make note of is that we found these large regions of, these large undulations in the outer membrane. So large changes in membrane architecture, which were not observed in the untreated E. coli cells. This led us to believe that pentamidine was directly targeting the outer membrane, particularly the LPS component of the outer membrane. So we hypothesized that if we typically get the nice synergistic reaction between pentamidine and rifampicin, if pentamidine is in fact binding LPS, we could add exogenous excess LPS to this assay, which should mop up all the pentamidine and alleviate this synergistic interaction. Indeed, with the addition of exogenous LPS, we see we lose our nice staircase checkerboard and are left with very little interaction between pentamidine and rifampicin. So we attribute this to pentamidine binding LPS, getting mopped up, and now unable to interact with the outer membrane LPS. We also conducted an LPS binding assay, which is a good way to quantify just how tight pentamidine is binding to LPS. So how this assay works is the LPS is bound to a quenched fluorophore that is displaced with increasing concentrations of pentamidine, releasing this fluorophore and causing fluorescence. So what's nice about this assay is it gives us this quantitative IC50 value. Our next step was to see if this synergistic interaction existed beyond just lab strain of E. coli. We tested a number of clinical isolates and a number of different bacterial species. In general, most species tested were susceptible to a combination of pentamidine with rifampicin. An exception where we saw no strains of Morganella or Proteus susceptible, and only 20% of Pseudomonas. Notably, 80% of strains of Acinetobacter tested were susceptible to our combination. And in performing these assays, we observed that Acinetobacter in particular was highly sensitive to the synergistic activity of pentamidine. So here we have Acinetobacter baumannii in a checkerboard assay of pentamidine against the gram-positive active antibiotic novobiocin. And we see a steep staircase in our checkerboard, showing us that this is a very potent synergistic interaction. This was encouraging, which led us to conduct in vivo studies on the combination of pentamidine with novobiocin. So our in vivo model is an Acinetobacter systemic infection in which animals reach endpoint in under 24 hours. We treated animals with either novobiocin alone or pentamidine alone. And what we found is that novobiocin at this concentration was insufficient to have any impact on survival. Pentamidine at the concentration used rescued 1 out of 10 animals, so about 10%. In contrast, if we look at the combination of pentamidine and novobiocin, we were able to rescue 100% of animals in this combination. So notably, the concentrations that we used are about 1/5 the human equivalent dose, which suggested to us that our combination, we might be able to dose spare moving forward into human patients. So we were encouraged by a number of things of pentamidine as an antibiotic adjuvant. It potentiated multiple gram-positive active antibiotics, and it did this against a broad spectrum of different gram-negative bacteria. And most importantly, we observed potent in vivo activity of our combination of pentamidine with novobiocin. So this led us to pursue a repurposing approach. As pentamidine is already used in the clinic, we thought this would be easier to have it approved for use as an antibiotic adjuvant. However, as we look for funding to help advance pentamidine into further preclinical studies and hopefully phase 1 trials eventually, we were met repeatedly with two major areas of concern, first of which is intellectual property. So an in-depth literature search into pentamidine shows that there's been a massive body of research conducted on this molecule since its discovery in the 1940s. And while we've not observed any prior art or any literature suggesting pentamidine's use as an antibiotic adjuvant, the excess of prior art we found would make it very difficult to receive a patent for this application. So this limits our ability to raise funding away from any private investment to more so a call for public health and government funding initiatives, and those overall proved unsuccessful. Additionally, another concern that we were met with is pentamidine is known to have toxicity, particularly to be nephrotoxic, as well as pentamidine is known to have an impact on herd channel trafficking. It causes QT prolongation, which can lead to negative cardiac side effects. So impacts on the herd channel in particular can be a deal killer when trying to move forward with a compound due to safety concerns. So this led us to move away from a repurposing approach and into more of a development of a novel pentamidine analog. So we hope to build a better pentamidine. And to do this, we were luckily able to collaborate with the Broad Institute, a group at the Broad under the direction of Dr. Deborah Hung, had received funding from a US government funding initiative, CARB-X, which looks to provide funding in taking antibiotics or lead molecules from discovery through preclinical experiments. So our strategy was to develop novel analogs. So this would take care of the intellectual property concerns. Our indication we chose to focus on was Acinetobacter baumannii due to our potent in vivo activity previously observed. And we hope to either increase potency or reduce toxicity of pentamidine. So our workflow is we look to modify the scaffold of pentamidine. We refer to this as our P series of molecules. We looked at modifications of the hydrophobic linker region, the aromatic rings, as well as changing the positive charges present in the amide groups. So initially, we developed a number of different analogs called our Series 1 analogs. We prioritize these based on efficacy in a checkerboard, as well as their ability to bind LPS. The prioritized analogs from Series 1, we then further developed into our Series 2 analogs. And with these, we once again tested efficacy, but then looked also into toxicity and their activity in vivo. So increasing potency of pentamidine potentiation proved relatively successful. In our first series of analogs, we developed the analog P2. And here you can see that in an Acinetobacter baumannii checkerboard, combining our analog with novobiocin, we see this very steep staircase pattern, which tells us that we have very high potentiation of novobiocin by P2, even more so than pentamidine. Our second series of analogging gave us P4, which you can see we've even been able to further reduce the concentrations of our of P4 required to sensitize to novobiocin. So we're very happy with the levels of efficacy that we were able to obtain after only a few rounds of analogs. We also tested these analogs for their LPS binding. So in this assay, the higher the number the less efficient it binds LPS. So we can see that we're increasing LPS binding efficiency by almost tenfold in each iteration of our analog development. And it was interesting that efficacy in our checkerboard correlated so well with increased LPS binding affinity. However, we reached quite the roadblock as we moved into our toxicity assays. So here we're looking at a HepG2 mammalian cell line assay. We're looking for what concentration causes inhibition of 50% of the cells in this assay. So here, a higher number means a higher concentration of drug required, which means a compound is less toxic. So we see that in our analogs with increasing efficacy, we see also increasing toxicity. This was particularly worrisome as early tolerability studies in mice showed that while pentamidine has some moderate display of symptoms, it's moderately tolerated. However, both of our analogs were poorly tolerated in the animals, irrespective of dose. So we looked to change our plans, looking to move away from emphasizing so much on efficacy and moving towards a focus on reduction in toxicity. We went back to our series 1 analogs and we prioritized those which had low levels of toxicity and efficacy that seemed in the range of what we'd observed with pentamidine. We then once again developed new analogs based on these prioritized leads and conducted our efficacy, tox, and then in vivo activity assays. So for a number of rounds of analoging, we eventually stumbled on P35. So if we look at the efficacy of P35 in synergy with novobiocin, we see a very similar checkerboard pattern as that of pentamidine. In our LPS binding assay, we do see some increase in LPS binding, although what was most important is in our toxicity assays, we see the concentration of P35 is much higher than pentamidine in our HepG2 inhibition assay. Additionally, in our tolerability studies, we found that while pentamidine was moderately tolerated, we did not observe any serious side effects of P35 and it was highly tolerated. So this gave us the confidence to move forward into in vivo studies. We used the same model that we had tested pentamidine against, pentamidine, or P35 in this case, combined with novobiocin. Here what we found is that all animals, once again, succumb to the infection under 24 hours, irrespective of treatment, either with P35 or novobiocin. The combination of P35 with novobiocin was able to rescue 100% of animals. So it worked at least as well as pentamidine at this concentration and displayed less negative side effects. So in our collaboration with the Broad, we've been able to move forward into further preclinical investigations into P35. So there's a lot of information here, but what I just really wanted to point out is that we move forward with some in vitro and in vivo parameters, including things such as compound stability, drug-drug interactions, and protein binding. In this case, everything came out very positive. We also looked into mouse PK data. P35 is similar to pentamidine in which it's rapidly cleared out of the animal. So improvements in pharmacokinetics are an area which we could maybe hope to advance with analogs of P35. Also very important, and something that I noted previously, hERG channel inhibition. We compared pentamidine with our P35 analog in 2 assays looking at effects on the hERG channel. So in a hERG channel inhibition, we did not see significant inhibition of the hERG channel by pentamidine or P35. But what's most encouraging is that in a hERG channel trafficking assay, pentamidine has an IC50 value of about 1, whereas we were not able to observe any hERG channel trafficking changes in the highest concentration of P35 testing. So overall, moving forward, we're looking to perform more chemistry on P35 scaffolds. We want to improve the PK of this molecule, as well as potentially extend its spectrum beyond Acinetobacter baumannii. So this work has been a joint effort by McMaster University and the Broad Institute. There's a lot of people who've worked on this project, some of which I've just highlighted here, my supervisor, Dr. Eric Brown, Dr. John Stokes, the original discoverer of pentamidine, Dr. Maya Farah, Dr. Sean French, and Dr. Brian Coombs for helping the in vivo work. The Broad Institute, we'd like to thank Dr. Deborah Hung, Dr. Katie Lee, Dr. Brian Hubbard, and Dr. Michael Serrano Wu for their assistance in this project. So thank you.
Thank you, Craig. That was really cool story. I hope it makes it all the way.
Yeah, me too.
All right, so there's one question, but I'm gonna reserve the questions until the end. So we'll move now to Fiona's presentation.
Are you seeing my slides?
Not yet.
Okay.
Don't you love technology?
I just have to get rid of my slides somehow, and then I can get back in, hopefully, to it, but it ought to be. Just give me a moment, okay? Maybe answer the question.
Sure. All right, I will read Craig the question if you want to pop back on. So the first question is from Hindra. Given the pentamidine structure, were there any predictions on how various structural modifications would reduce its toxicity?
Yeah, so I'm not a chemist, but I know that one thing they focused on a lot was the hydrophobic linker region. It's known that long chains of carbon can be toxic, and we found that additionally increasing the hydrophobicity with increased length of our carbon chain was able to increase toxicity. Unfortunately, this also correlated with increases in potency. So that's an approach that we tried early on, but we eventually had to abandon.
Okay, thanks. There's one other question from Jerry, but I'm going to hang on to that one until the end, now that Fiona has gotten her presentation to cooperate. All right, take it away, Fiona.
Okay, thank you very much. So what I thought I would talk about today was really a story of designing and implementing a phase 1 clinical vaccine study. This, I think, has a lot of timely relevance as we're hearing so much about COVID vaccines. But I want to take you back a step to the research that we've been doing in collaboration with Dr. Zeng and his team on a tuberculosis vaccine. So the objectives of my talk are to understand the stages of the clinical development of a new vaccine candidate, and then review what Health Canada requires from a sponsor for a clinical trial application. That will include the preclinical results, everything to do with manufacturing, the planned study design, and the qualifications of a team. And I've just put in italics there, from a sponsor. Most of the clinical trials we hear about for vaccines are sponsored by pharma, and they have multiple resources to actually move these clinical trials from a phase 1 after the preclinical data right through to phase 3 and regulation. We at McMaster really don't have those resources behind us, and it's actually the university that is the sponsor of our clinical trial. And if Jonathan's on the line, that also comes with responsibilities. I will highlight the challenges and the rewards of doing this towards the end. So clinical trials can be divided into phase 1, 2, 3, and then phase 4. They're all based on initial preclinical data and then a phase 1 study, generally with 20 to 80 participants, is really the first opportunity for the drug, the vaccine, to be tested in humans. The general goal of a phase 1 study is to evaluate the safety of it, and in vaccines also to get some preliminary data on the immunogenicity of it. Generally, as I'll talk about later, we look at a healthy adult population, not necessarily the population that may be most at risk of the disease, to enroll in a phase 1 study. They're generally open labeled, and because they require probably a close amount of monitoring, they're usually done fairly close to, as we are here, a tertiary centre where you can monitor participants very closely for any adverse events. We then move on to a phase 2 when there is some satisfactory data from phase 1. This is sort of where many of the vaccines are sort of divided into a go or no-go. The funds to take a clinical trial right through to phase 3 are considerable, and so it's important that there are criteria early on to decide whether a product is going to be a go or a no-go. And we're particularly interested in this from our TB vaccine work. Generally, the study would be big enough to provide some statistical power with an understanding that there'll be some clinically meaningful outcomes, both on safety and efficacy. And then if you make it through the phase 2 study, where you may have included some different populations, such as those that are older or those with chronic disease, then you will go into a phase 3, which is where we hear many of the current COVID study vaccines are, which is really in some ways quite exciting. This really is looking for both, again, safety and efficacy in a large population, a large representative population. And so this often is where some of the less common side effects may be identified. And these data are the ones that are required for regulatory approval. Phase 4 studies are generally after the drug has been approved and looking for more registry of the use of the drug to pick up anything that is untoward or unexpected. So I'm going to talk a little bit about our tuberculosis vaccine program. Many of you will have heard discussions about this before, but for those of you that this is new to, we are studying a recombinant human serotype 5 adenovirus vaccine. In this, it is replication incompetent, and the primary antigen, immunogenicity antigen, AG85A of tuberculosis, has been incorporated into the adenovirus vector backbone. You'll have heard quite a lot about AD5, because this is the backbone of some of the other vaccines for COVID that are currently being discussed, including the Russian one and the CanSino one. But I think it's very important, and this is perhaps quite timely, for us to honour the work of one of our really emeritus faculty, Dr. Frank Graham, who probably more than 45 years ago discovered here at McMaster the AD5 vector, and all of the work that has been done since then really has rested on his shoulders. So we, I think, can be proud in lots of ways that AD5 really was came out of McMaster with the work of Dr. Graham. Now, I mentioned that one of the important things is moving from a preclinical data to the first in human, and it really is vital for robust preclinical study data. I'm going to talk a little bit about how these requirements may be being modified in the era of COVID, but really to move into humans, you need to be very confident of your animal data. And this is just an illustration of the preclinical work that has been done mostly in collaboration with Dr. Zing and his team, as well as colleagues around the world, looking at initially murine studies, moving on to guinea pig, goat, bovine, primate, humanized mice, and then our phase 1 clinical studies. So really this is a long, long haul, or let's say this has been a long, long haul. We start off here, and you can see that this paper was published in 2006, looking at the effect of the vaccine on the protection against TB in a mouse model. Just to orient you, these were mice that were either immunized with BCG and then boosted either with BCG, the adenovirus vector given intramuscularly, or the adenovirus vector given intranasally. And you can see, based on the colony-forming units in the lung of Mycobacterium tuberculosis, that the BCG-primed with the intranasal administration was the most protective. And so this was in a mouse assay, a mouse experiment done in 2006. When we move on to guinea pigs, in this situation, we're looking here at survival. So again, we've got mice that are given BCG to prime them, followed up with either the intramuscular or the intranasal vaccine, and then challenged with TB. So here we're looking at an efficacy model with challenge with tuberculosis, Mycobacterium tuberculosis. And you can see that survival is greatest with the group that received BCG, followed by the intranasal administration of adenovirus vector as compared to the IM one. And looking again at a cattle model, you've got the same with BCG primed here with the adenovirus vector. Looking here at two outcomes, either the lung pathology, representative of mycobacterial infection, and you can see that the lung pathology is the lowest here in the group that received BCG priming with the adenovirus vector. So once we've got some excellent preclinical data, which I think you would all concur with, having reviewed really just a very small selection, the next part of proceeding with a phase 1 trial is to look at the chemistry and manufacturing control. And we are extraordinarily fortunate here at McMaster to have the facility at the Robert E. Fitzhenry Vector Laboratory up there on the 4th floor, where we have and continue to make clinical grade vectors. And so in the submission to Health Canada, they are looking at all the components of the manufacturing of this product to ensure that it meets all good clinical practices. And the clinical lot requires extensive testing for safety, purity, strength, potency, before we can actually proceed on. And as I mentioned, we are extraordinarily fortunate to have such a skilled team in our institute. So at this point, I have to shout out a big thank you to Jerry here, because we had no money, and so we heard from Craig how it can be a bit of a challenge when you have no money. Our first phase 1 clinical trial was started with a $10,000 grant from the IIDR to actually put in place our first clinical trial agreement. We needed help with the regulatory requirements, and so we had no funding to actually do a phase 1 clinical trial at this point. But Jerry, and I know this is something that happens to other groups as well, we got our first leg up with funds from the IIDR. So again, thank you, Jerry. So our phase 1 trial design, our first study that we did probably around 2010, looked at taking 12 BCG naive participants and 12 BCG positive volunteers. We had an age range of 21 to 25 years. We gave them all intramuscular adhuman 5 AG85A, one dose here at 10 to the 8 particle forming units. We screened our participants to make sure they were healthy. We included negative HIV, a normal chest x-ray, no evidence of latent TB, and then followed these participants after immunization with blood tests looking at antibodies as well as immune responses to the vaccine. So remember, this is the first in-human study that we had done with this product. And we were able to demonstrate that our vaccine did induce robust T-cell responses. And this was despite many of our participants having anti-Ad5 antibodies. And you can see here the immune responses. I've just illustrated the cytokine responses in the percentage of CD4 cells, the BCG positive group and the BCG negative group at 2 weeks over to 24 weeks. So the exciting thing about this was that we actually had seen that the vaccine did induce immune responses. We were also, because safety is one of the most important things, looking at a number of good safety outcomes. So as one would expect with an injection, there are injection site reactions, and they were common. There were some participants who complained of a headache, fatigue, malaise. Many of our first volunteers were people actually from our group. And so it's important as we're recording these to make sure that we don't neglect any type of adverse events. And that's part of the very systematic way that we go about collecting adverse events or any adverse event that will occur. Arthralgias, nausea, fever. All of these, however, were judged either mild to moderate, and they all resolved within 24 hours. So we were quite confident that this was a safe and well-tolerated vaccine. So as you'll recall from the initial preclinical studies that I presented, the intranasal route of the vaccination was going to was demonstrated in the animal models to be more effective in preventing tuberculosis. And so many of you will have asked the question of us, well, why aren't you giving it into the lungs intranasally or by some route mucosally? And so our next challenge, after we had completed our first phase 1 study with the IM administration, was to design the next study, which is to administer the vaccine by aerosol. And so what I've done here is I've listed a number of many questions that come up as we're starting to think about designing the study. I don't have time to go through all of the aspects of the study design, but I can just mention some of the important criteria that we had to consider as we were going forward. We, of course, wanted to have healthy volunteers. What should our inclusion and exclusion criteria be? We were giving people a vaccine that we were going to administer by aerosol into the lungs. So we were concerned about lung health in terms of any unexpected toxicities. It's very easy when you've got a mouse, you can just drop the vaccine into the nasal passages. But our challenge was, how would we administer the vaccine by aerosol to humans and ensure that we would get particles small enough that would get into the lungs? The challenge with many phase 1 studies, and in fact, many phase 1 studies are designed to establish this, is what dose should we choose? And that is always challenging, somewhat arbitrary, but something that is very important in terms of discussing and also modifying as going forward. One of the things about Health Canada is that they require all your vaccines, all your drug products to be appropriately labelled. So knowing what the regulations are around product is very important when you're moving into a phase 1 study. And as I always highlight in any discussions, we would be potentially open to a Health Canada audit where they would come and look at every stage of our study. So when you're conducting these studies, when you're designing these studies, it's good to remember that keeping on top of all the regulations is not only wise, but it's also very necessary in terms of what the implications could be if you did it wrong. We were giving the vaccine by aerosol. Would there be any infection control precautions? Now, adenovirus vectors have been used very safely, and I think at this point we are quite happy about continuing with the precautions we've put in place. But at the beginning, there were a lot of concerns about adenovirus and the effects that it might have on the environment. Sample size is always a challenge in phase 1 studies. How many participants do we actually need to enroll? Time is of the essence, cost is of the essence, but if you don't have enough participants, you won't get any outcomes. And it's very hard with a phase 1 study to really know what your sample size would be. So many of them are designed sort of as a pilot study with the outcome getting clinical data, getting toxicity data that can then be used to correctly calculate a sample size for a phase 2 study. Knowing what side effects you might expect are important. Establishing and confirming the role of a safety monitoring committee. And I'll talk just a little bit about stopping rules, because one of the concerns, of course, with any study is that something might go wrong, and how are you actually going to manage that risk in your research. So I've sort of titled this, "It Takes a Whole Village to Raise a Child," but the team that we have at McMaster that were involved with this is representative of many departments across the faculty. And for those of you who are young at McMaster, perhaps for the first time, the real benefit of this place is recognizing all of these amazing collaborations that happen very seamlessly, and I have to give a shout out to the collaborators on this study. We were particularly fortunate to have the expertise of Dr. Dolovich, who is probably a world expert on aerosol delivery. The laboratory, Dr. O'Byrne and Gavro's laboratory, where we do the bronchoscopies, was a vital part of what we're doing. And of course, the scientists within the department analyzing all of the samples. So I'm just going to take you through very quickly the inhaled aerosol delivery system. We use the AeroNeb, which is a nebulizer that is recognized in clinical medicine as providing a effective and predictable particle size. This looks like a little bit of a homegrown system, but this was how we were establishing what the particle size that this nebulizer would provide of our vaccine. And you can see here that the particle size is within the range of 2 to 5 microns. And this is just at that level of the 15 to 20 generations of the small airways, so really quite distally in the lung. And by doing a number of different experiments with Dr. Dolovich, we determined the volume we would need, how long it would take, what proportion of what we put into the nebulizer would actually reach the airways, and particularly the size of the particles, guaranteeing that it would get into the lung. This is the system that we have set up currently to administer vaccine to our participants. It does look a little bit homegrown, but we're looking to involve others so that we can simplify some of the administration of it as we move forward. So one of the things about a study like this is making sure that you have healthy participants. And any phase 1 study is going to have a long list here of exclusion criteria. So many of the phase 1 studies really exclude anybody who has any acute or chronic illnesses. Because we were dealing with the lungs, we needed to have people who had healthy lungs. We weren't able to have cigarette smokers, and Health Canada wouldn't allow me to have any cannabis smokers because of the effect that that may have on the lungs. You could have had a history of cannabis smoking as long as you promised not to smoke it during the study. And because part of the measures we were doing was looking at the immune response in the lungs, patients were having a bronchoscopy, a bronchoalveolar lavage, and so any contraindications to that procedure were also part of the exclusion criteria. We had some age cutoffs of 55, but we can discuss whether that is relevant going forward. So this appeared, and it's actually a long time ago now, 2006, but the headlines are quite dramatic. London drug trial catastrophe, collapse of science and ethics. And here's a young fellow, I nearly died in a medical trial. He thought he found a way to earn some easy money, but. And when this study was reviewed, when 6 of the young volunteers became violently ill minutes after taking the drug, there were a number of important caveats that really weren't followed, and which now, of course, we build into our clinical trial and should have in those days as well. Their preclinical data was very sketchy at the best of times, and really there was no good published evidence that had been peer-reviewed of the efficacy of the drug. And the other thing is that they immunized, or that they gave the treatment to all 6 participants at exactly the same time on the same day. So what we do is, when we take this into account when we're looking at how to design our clinical trials. And so you'll see here in our protocol, under the study procedures, we will first vaccinate 2 subjects. Assuming no grade 3 or 4 adverse events, they're the more serious ones, or any serious adverse events, at least possibly associated with vaccine, who've been followed for 2 weeks, we will proceed to immunize the rest of the subjects in this group. So these are built into the protocol and really required by Health Canada. We have a safety monitoring committee, independent from the investigators. It's in infectious diseases and respirology. And we use them to make decisions about moving to the next or higher group, depending on what the study design involves. So we will expect the Safety Monitoring Committee to look at the data that we provide after all the subjects have received a dose of vaccine and been followed for at least 4 weeks. They've reviewed all the safety data. No SAEs that are probably or definitely related to it have occurred, and no dose-limiting toxicities. So this is built into the protocol. You can't make it up as you go along, and I think gives confidence to the quality of the type of research that you're doing. Dose-limiting toxicities, again, defining those in advance. So if any participant has experienced a serious adverse event, either possibly, probably, or definitely, or similarly, there is grade 3 or 4 AEs within a dose level. So these things, as I mentioned, are in the protocol. Now, we are currently about 2/3 of the way through enrolling the TB vaccine administered by aerosol. We are still looking for volunteers, so I've just put up the poster here. We have an email, we have a Facebook page, we can be found through the IIDR, and so any volunteers who would be interested, who have had BCG, we would be pleased to discuss it with you. And I should say that with the participants we've enrolled, there have been no serious adverse events, and there have been no pattern of adverse events that would be of concern. So this really is just an opportunity, the story that I've told you, to just introduce at this point McMaster's COVID-19 vaccine project that I'm very fortunate to be working with Zoe and Brian on. And I've mentioned a small army of researchers. This again is a research proposal with many people contributing to it. We're looking at a bench-to-human development of a safe, effective aerosol vaccine. And those of you who have been familiar with the work that's coming out of Zoe's lab will be very familiar that we sort of look at a 3-pronged approach to respiratory mucosal immunity. This is similar to what we're looking at for tuberculosis. Trained innate immunity is getting a lot of press at the moment, and this is, I think, a very important component of the aerosol vaccine strategy using a vector, including neutralizing antibodies and T-cell immunity. So it's really this three-pronged approach to immunity that a vaccine, a viral vector vaccine administered by aerosol can likely produce. We'll be using both an Ad5 and an Ad-chimp viral protein. Rather than just using the spike protein, which many of the others are doing, it will have a multivalent design, the nucleocapsid and the polymerase as well. And as I've mentioned, look at the respiratory mucosal route of vaccination. This is just showing the and the chimp, which is an Ad-chimp 68, again with the spike nucleocapsid and polymerase. So I pointed out the fact that the first study we did in mice was in 2006. Here we are intending to have this study done in probably 18 months. We've stage of doing the small animal efficacy. We're working on the GMP production, and based on a lot of the work we've already done on a clinical inhaled aerosol study for TB, we hope to very quickly be able to do a clinical trial with this product in another phase 1 study. So we've come a long way from Jenner and his phase 1 study here of smallpox vaccine, but I think that the next number of months are going to be very exciting for us, and it's been a wonderful opportunity for a clinician to be involved with this cutting edge research within the department. So I will stop there. Thank you.
Thank you. That was terrific. Drug and vaccine development takes a lot of time, a lot of money, and a lot of people and their intellectual input. I also appreciate that you shouted out Frank Graham there. It's hope for people doing research that, you know, even if it doesn't seem like it's going to be taken up immediately, there's always the future. Okay, I'm going to read out a few questions. We have, if you can stick around, I think Christy booked about 15 minutes. So I'm going to ask a question for Craig. What's the thinking now on pairing your new version of pentamidine with an antibiotic? So which antibiotic?
Yeah, so that's something we've thought a lot about. We've had some pushback from our use of novobiocin as it was pulled from the market in the early 2000s. So our dream would be actually linezolid as an antibiotic partner, but it's mostly efflux that makes gram-negatives not susceptible to linezolid. So we don't see great potentiation with our analogs, unfortunately. We've also considered moving forward with macrolide antibiotics. We see pretty good synergy with things like erythromycin and clarithromycin.
Cool. Okay, the next question is also for Craig from Mike Surratt. He says, great work, great to see the pentamidine story moving forward. So 2 questions. First, do you know the mechanism of resistance in the Morganelliaceae? Is it the LPS structure? And second, at least with pentamidine, the combinations take broad spectrum gram-positive and make them into broad spectrum gram-positive and gram-negative antibiotics. Independent of the potential downside of broad spectrums, do you think this might accelerate resistance?
Okay, yeah, so the first part, both Morganella and Proteus have much different LPS structure than E. coli, so I think you're correct here. it seems that anything that's intrinsically polymyxin resistant is also resistant to the action of pentamidine. Interesting though, if you have acquired polymyxin resistance by MCR1, we still see potentiation with pentamidine. So I think that could probably suggest that it might be the amino arabinose decoration, which I think is present in Morganella and Proteus on their LPS structure, is something that we can't overcome with pentamidine. But the phosphoethanol decoration conferred by MCR1 or other types of polymyxin resistance can still be overcome by pentamidine. For the second part of your question, that's also something that we've received some pushback on, is that there's actually a lot of pre-existing resistance in gram-negatives to gram-positive antibiotics. This was the focus of a paper which our lab just published a few weeks ago. In terms of how the interaction between outer membrane perturbants and these mechanisms of resistance work, it seems to be broadly, if you're an antibiotic inactivation enzyme, you can think of something like MPH enzymes for macrolides. In most cases, the use of a potentiator and a macrolide can overcome this resistance. We think it just floods the cell faster than the inactivation enzyme can work. But in terms of target modification enzymes, so we are concerned about moving gram-positive resistant elements into gram-negatives. In fact, in many cases, they're already there. So it would likely increase this type of resistance downstream.
Okay, thank you. The next question is for Fiona. So from Mohammed, he says, in regards to the nasal administration, were respiratory individuals excluded from the study? And if not, were there variation in results between respiratory versus non-respiratory participants?
Well, I should clarify to begin with, we administered it by aerosol directly into the lungs, so it was inhaled into the lungs. And in our phase 1 study, we did pre-screening with pulmonary function tests, including a DLCO. We really wanted people who had totally healthy lungs, so we didn't have any participants who had any underlying lung disease. That was one of the exclusion criteria. Interestingly, when we're thinking about COVID-19, which does affect those with underlying pulmonary disease, cardiac disease, and the elderly, it'll be interesting to think through how we may enroll in our study those participants in whom the risk is greater, but whether we would put them in a phase 1 study is unclear. So I think we'll move slowly into the more high-risk group once we've established safety in the healthy participant group.
Okay, thanks. The next question is also for Fiona from Mike Surratt. So he says, Fiona, a naive immunology question. Would mucosal immunization in the nasal passages provide sufficient protection, or is initiating the immunity in the lung necessary? Could aerosol delivery to the lungs be avoided?
I think that is an important question, and there have been studies looking at the nasal administration. There is an influenza vaccine that is administered nasally. One of the slight concerns we have is that there has been some documentation of the viral vectors moving up through the olfactory tract to the brain, and so that is a relative caution about using the nasal route, but clearly there are some precedents, and again that is something that is being explored.
Okay. The next question is from Jerry regarding anti-AD5 antibodies. Are there any concerns about different levels in the target patient group? For example, not Canadians. So I guess in the more global perspective.
Yes, well, this actually was something that came up, was that AD5 antibodies are, I'm going to say, relatively uncommon, or let's say about 60%, say in a North American population. They'd be closer to 90, 100% in a sort of African developing world population. And so a lot of early criticism of the AD5 vaccines for COVID have been that these are sort of Western world vaccines and wouldn't be applicable in a developing world setting. This is indeed part of the reason why we're exploring the AD-Chimp 68, because there's an expectation that neither the developing world, if that's the right word for it now, or our Western societies have pre-existing antibodies to that. So I think the point is an important one, and one that's being taken into consideration as we move forward with our vaccine and the chimp vectors.
Mm-hmm. Okay, thanks. The next question is for Dr. MacNair from John Stokes. Actually, two questions. What's the frequency of resistance to P35, and does P35 have any intrinsic gram-positive activity?
So for the P35 resistance, we don't observe really any antimicrobial activity of P35. So we haven't tested resistance of P35 alone. If you're talking about the combination of P35 with other drugs, that's something that also, unfortunately, we haven't tested. Once we hone in on an antibiotic partner, we'll definitely be conducting those tests. In terms of intrinsic gram-positive activity, it's an interesting question because, yeah, pentamidine has some gram-positive activity, and we see similar levels of gram-positive activity in our P35 analog, about 8 micrograms per ml. MIC in P35. So it does in fact have some gram-positive activity as well.
I have sort of a related question about potential effects on the microbiome of something like a sort of more generic permeabilizer like that. Do you worry that it's, you're going to pair it with an antibiotic, but is it going to potentiate the entry of other molecules into cells where they wouldn't normally get in, and is that going to be a problem?
In terms of bacterial cells?
Yeah.
Yeah, you're basically taking a broad spectrum, you're turning a narrower spectrum gram-positive treatment into a completely broad spectrum approach. So yeah, any gram-negative parts of the microbiome are likely to be targeted. There are some species which are intrinsically resistant to polymyxins, so they'd probably be resistant to the potentiation, but we don't know exactly how important that is or what percentage of the microbiome those make up. So it would probably wipe the microbiome like a standard broad spectrum, unfortunately.
Okay, well, that was all of our questions. So I want to thank you both again for giving us the whole spectrum from screening all the way to getting something in a human, and how amazing of a process that is. On the subject of vaccines, before we leave, I just want to say, everybody, get your flu shot. They're now available, and I'll be going today. So thanks again, everybody, for joining us, and I will see you next month.